So good afternoon, everybody, and welcome to the 5th webinar of the VALITEST series on the organization of test performance studies. So VALITEST is an EU-funded project undertaken by a consortium of 16 partners and built to improve the reliability of diagnostic tests performed in plant health laboratories across the European and Mediterranean region. And if you want more information on this project, you can go and check our website. This series is about the organization of test performance studies. So for the one that haven't been attending the previous one, test performance studies or interlaboratory comparisons that aims at evaluating the performance of diagnostic tests. And during the series, VALITEST partners are presenting the different steps of the TPS workflow through different videos, webinars, and training activities. So today we will discuss about the production of reference material with René. So the presentation will be recorded, and a link to the recording and a link to the PDF presentation will be provided on the VALITEST website. So today our speaker is René van der Vloot. So René was the leader of work package 3. He is a senior scientist and plant virologist, head of the plant virus group at Wageningen Research in the Netherlands. And he is also a professor of ecological plant virology at Wageningen University. So he has over 30 years of experience, both in biological and molecular research on plant viruses, and he is responsible for research projects on the identification and characterization of and epidemiology of plant viruses. And he participated in many EU-funded projects, including Q-Collect that he might mention during the webinar, but also, of course, VALITEST. So, René, I will now give you the floor.
Okay. Thank you, Charlotte. Yeah, welcome all. Well, Charlotte has already introduced me, so I will then continue with the webinar. Obviously, this was not my own work, my fully own work. I mean, Tanja Dreo and Anne van Diepeningen from Wageningen and Tanja from the National Institute of Biology in Ljubljana, Slovenia, were also very much involved. And they participated with me especially in work package 3. And today we will discuss, let's say, more on the production of reference materials on test performance studies. There will be a few polls, so questions in this webinar, and Charlotte will take care of that. And you are kindly requested to answer simple questions, either press yes or no, or pick one of the 4 answers that you are, let's say, presented. Please respond as quickly as possible, because it will help us to get an idea, and also you to get an idea about who is actually now participating in the webinar. At the end, we'll have plenty of time for discussions, so please put them in the chat or remember them, and then we can discuss later after I finish my presentation. So I would like to start by saying what we're going to do within this webinar. We're going to discuss on what is biological reference material and why do we need it. What different types of biological reference material are there? What are the criteria that biological reference material should adhere to? And how do you actually produce it, right? And as I said before, we will have a few questions in between to get an idea. So I would like to start off with a first question, and that's a very simple one. And please answer as quickly as possible. Do you actually use reference material, or do you not use reference material? Just click yes or no on your screen.
Yeah, so we have about 90% of the people that have answered. I will just wait a few more seconds.
Okay, that's good. Nearly 100%.
Yeah. Okay, so it's not changing so much. So I will close the poll and share the results. 94% of the people answered that they use reference material and 6% that they do not.
Yeah, so 94% of the people who answered actually use reference material, and then 6% does not use reference material. So that's very good. And then, whoops, where's my mouse again? Here. Then the second question is what lab are you actually from? So we have 4 answers. Obviously, it won't cover everything, but it's either from an EU or national reference laboratory, either from an official diagnostic laboratory, from a private commercial laboratory, or basically other, which means none of the above. So if you can answer that question as well.
So we have about 85% of the people that have answered. I will wait a bit longer.
Yeah.
Okay, so it's not changing so much. 87% have answered. I will close the poll and share the results. So we have 67% of the people that are from EU or national reference laboratories, then 16% that are from laboratories performing official diagnostics, 9% from private or commercial laboratories, and 7% other.
Yeah. Okay, so thanks. About nearly 90% people, nearly 90% also answered. So we have about 67% from EU or national reference laboratories. We have 16% of official laboratories, 9% from private, and 7% of others. So really good, nice to know. So I think we first need to answer the question is, why do we actually need reference material? And there are a number of reasons. Most of you already work with reference material, so some of these reasons will sound very extremely familiar for you. A very important one is reference material provides essential traceability in plant health testing. It allows you to, let's say, perform internal and external quality checks, and you know what material that you've been using and why. You can use it also for validation and verification of tests. So and there's an APO standard on that as well. And reference material is often used in inter-laboratory comparisons or test performance studies. And you can also check out the APO standard 122 on that, but you're probably already familiar with that. Charlotte already mentioned VALITEST. So a little bit of background. VALITEST is a big EU project on the validation of diagnostic tests. As Charlotte said, it has over 16 participants in it. And one of the work packages was work package 3, and that was on the quality assurance of reference material for validation purposes. And within that work package, we had 2 main tasks. First one was to identify criteria for reference material production. And the second one was to design a standard operating procedure for the production of reference materials. Obviously, the material is quite diverse, so you cannot design one standard operating procedure, but at least a framework. And that was done. More information on that later. So if we know why you want to use reference material, then you also have to ask yourself the question, what is reference material? And as it is defined by the ISO organization, it is any material which has been established to be fit for its intended use in a measurement process. And obviously, there are additional, let's say, criteria there. I'll add a pointer, which is a bit easier. There are additional criteria here, like it needs to be sufficiently homogeneous or stable, and there are other criteria, and we will get to them later. But basically, you know, it has been established to be fit for its intended use. That's a very important, let's say, thing to remember. It's also important to realize that reference material is basically a very generic term, because it's a quite broad term, and we'll get to that later. But its properties can be either quantitative or qualitative. So about, qualitative, yes, no, and or quantitative, and that is how much. Apple also has a definition on reference material, which is slightly different from the ISO, but basically not that much different. Again, it is material that is appropriate to the test and the diagnosis being performed. And Apple expands a little bit on that definition by giving more, let's say, clear and detailed examples, like it can be live cultures of a particular pathogen, infested plant material, DNA, RNA, right? But Apple definition also mentions that the reference material used should be documented, right? And it should be ensured that the material used is producing the features for which it was selected, and explicitly meaning there that if you use reference material, for instance, in the serological assay, you need to make sure, you need to know that it actually expresses the desired antigen that you are testing for, or for instance, that it displays specific physical features, if it's, for instance, used for morphological diagnosis. But where does reference material come from? Since it can be so diverse, it's very obvious that there will be really diverse sources. But in general, one can say that reference material is more reliable when it's sourced from an entity that can both certify the authenticity and the quality of the material. How you can do that, we'll get to that later, right? And Charlotte also mentioned the EU project on Q-Collect, and that project was focusing on the collections that are present in Europe, available in Europe, reference collections of plant pests and pathogens, with an emphasis on quarantine plant pests and, for instance, invasive plants. And one of the outcomes of that project is that there is actually a very limited number of collections that are of a high enough quality, right, to be able to really trustworthy and reliably provide material. So that is something that we need to take into account, especially when you, let's say, are talking about the sources of your material. What's also important to realize is that it's not just, let's say, the source and the type of material, but we defined within the work package 3 of VALITEST is that reference material is not just describing the material itself, right? Basically, it also encompasses the subsequent steps of preparing the material, because in many cases, there is not enough, for instance, of naturally infected material, or it needs to go through a number of processing steps to either make it into a suitable form or produce enough to be used as reference material. And again, that's something that we will come back later. This slide just shows you an example, where can it come from? For instance, an example from bacteriology. We've got bacteria from a culture collection, well characterized, well identified. And when you want to prepare reference material based on these on these bacteria, for instance, you can start growing them in culture. You can then prepare suspensions with, for instance, defined concentrations, and you can use that directly as reference material. But one step could also be to, let's say, mix it with plant material, right? And then prepare reference material based on purified bacteria together with, let's say, plant material. And that is one way of producing, but obviously there are other ways of producing reference material. And it just shows that based on the same type of material, you can have diverse preparations of reference material, and also from diverse sources. For instance, this is culture collection. You could also have, let's say, field material that is naturally infected with bacteria. So different types of reference material. Plant health testing is about testing for many, many different pests and pathogens, and a lot of those are actually not readily available in pure or in cultured form or in collections. And within work package 3, we decided to define, let's say, different types of candidate or biological reference material. And the most obvious material is naturally infested plant material. Like here, leaves infected with bluntpox virus. Yeah, but you can also have artificially infested plant material. So that is, for instance, an indicator plant or plant material artificially infected with a virus or with a bacteria or with a fungus, right? You can have spike plant material, like we just seen in the previous slide, whereby you take healthy plant material and you mix it with a bacterial suspension. You can sometimes use the purified or the isolated organisms. You can have total nucleic acids from a sample. So, for instance, a PCR reaction can be based on an RNA or DNA extraction from a sample. And there might be, let's say, your target organism in there, but there might also be other organisms in there as well. It can be purified nucleic acids, right? Or it can even be synthetic nucleic acids, like the use of G blocks and things like that. So there is a diversity there, and all these different types of materials can be used as reference material. And what possible sources can you actually really distinguish? Well, obviously, a field material is one source, usually naturally infested material. But that material, the use of that material raises questions on its identity, its purity. Are there any co-infections? Are there any pathogens in it? What do I know about the concentration of my particular pathogen, which I want to be there in that field material? You can have material from working collections. So working collections are collections that are present in individual laboratories or with individual researchers that people actually use in their own research, right? Or as material. Usually it's fairly well identified, but there can still be questions on whether it's really pure. What else is in there? Are there co-infections? Are we really sure about the concentration and stuff like that? We also distinguish reference material. That is material which is present from, or let's say that which comes from more well-established collections with certain levels of quality control and so on. That's usually quite well characterized. And we defined also a fourth category, and that is certified reference material. And certified reference material, according to its definition, is material that is let's say, coming from a collection which complies with the ISO 17034 norm. And that is a norm on culture collections. That is, let's say, difficult because it's a very strict standard. And basically, we don't really have any of those in Europe, except for DSMZ, the Deutsche Sammlung für Mikroorganismen und Zellkulturen in Braunschweig. And that is the only, let's say, culture collection that actually complies, at least to my knowledge, with this ISO norm. So they can supply certified reference material, but basically any other material coming from other collections is technically, at least according to the definition, you cannot call it certified reference material. But what is, let's say, a common thing in these possible sources is that going from field material to certified reference material, it has an increasing level of characterization and study. So again, Paul, I would like to ask you, where do you get your reference material from, right? Do you get it from field material, working collection, established collection, or really from the reference collection? Basically, in this case, that is an ISO 17034 recognized collection.
I'm just realizing that culture collection was not mentioned in your poll, René. So
Oh,
I
yeah,
guess it's what
that's
you
right.
call the.
It doesn't matter. It's okay. I changed the wording. Yeah.
So we have 70% of the people that have answered, so I will wait a bit longer.
Mm-hmm.
Yes, so we are at 85%, but it's not changing. So I will close the poll and share the results. So we have 40% of the people that answered field material, 69% working collection, 56% culture collection, and then 41% reference collection.
Okay, the people don't see this, huh?
Yes, they see it.
They see it. Okay, so yeah, so this is interesting because obviously people get material from diverse sources. It adds up to well over 100%. But a lot of people get their material from working collection and also from established, well, yeah, established or cultural collections. That's more or less the same. But still, 41% gets it from a reference collection. So that's quite a lot. Okay, interesting. We go back. We go on. So if you get your material, and especially if you want to start producing it for test performance studies, right, you have to ask yourself, is the reference material that I want to start using or supplying, is it really suitable? And one very important thing to start off with is you have to determine and define its intended use. You have to clearly define beforehand how and in what test format the reference material is going to be used. That needs to be carefully described and documented. So the type of study it's aimed for, the target pest or organisms, and also if there are any limitations on its specified use. For instance, if you know about background reactions in certain matrices or whatever for particular tests, right? And the properties that the reference material should adhere to, right, depend on its intended use. Again, you have different types and you have different types of tests, and both, let's say, determine what properties you actually need from your reference material. One of the very clear important things, obviously, is the identity of the reference material. It should be identified unambiguously at the relevant taxonomic level. You should use appropriate methodologies for that, and obviously you should use the up-to-date taxonomy. If you really identify your material, we suggest and we advise that you use at least two independent. My mouse is not really moving here. Yeah, you use preferably two independent tests, different biological principles, right? That's not always possible, and that's why we say preferably. Yeah, but basically you should try to identify your material with two independent tests. It should be documented, as I said before, and in case you prepare it by mixing, the identity of each component should be determined and again also documented. There are different, let's say, apart from identity, there are different criteria, and we identified a number of these in work package 3, right? And these define set of descriptors or criteria together form a basis for efficiently collecting and comparison of data on reference material. And your data should always contribute also to, let's say, the data being findable, accessible, interoperable, and reusable, so adhere to the FAIR principles. And the criteria are inherently linked to the intended use of the reference material. They may be test specific, so it's different for ELISA and it's different for a PCR. And again, it may depend on the intended use of your reference material, but they all should be defined by the producer. So this slide lists a number of these criteria, right? and we will go through them separately. First of all is the scope, the intended use. Identity is an important one. Traceability, commutability, it's a difficult term, but I will explain that later. Purity, homogeneity, stability, and assigned values. And if you want to learn more on the criteria here, I suggest you check out the website. On VALITEST, there is a page on work packages. Go to work package 3, and there you can download the deliverable which describes these criteria, and check them out and get more background information on them. But again, depending on the, let's say, the identity and the intended use of reference material, you should really realize that not all of these descriptors need to apply to a particular reference material. So scope, intended use. One of the criteria, it's a vital part of, of reference material, because you should define it before you start preparing it. You should ask yourself the questions, why do I need the data on my reference material? Yeah? What pathogen or pest are you going to test for? What method are you going to use? Will it be a morphological test, a serological test, molecular test? And again, are there any specific limitations to the use of the reference material, either depending on the method, the way it was prepared, or for instance, the matrix your reference material is going to be used in, or your test is going to be used in. Reference materials should also be traceable, right? And with that, we mean that there should be documented information on the exact source of the material. And again, you have two categories there. You have collection material, so it either comes from a working collection, a culture, or a reference collection. And basically that means that the information should be traceable to a specific specimen, or a specific isolate, or a strain, and if possible, information on the history of the maintenance and the handling in that collection should also be available. So it should be traceable. Where did the material come from? How was it produced? What was its history? And for non-collection material, and usually you can use the word field material for that, right? Specific metadata should be documented wherever possible. So, and that means, you have to know when, where, by whom the material was collected, from what plant species, what part, and whether or not it was showing which symptoms. And obviously, very important nowadays, you need to be aware and also document if there are any additional restrictions on the use of that material. And I specifically refer here to the Nagoya Protocol or specific material transfer agreements that might be, let's say, in place for that particular material. Okay, commutability. That's a difficult term, which often creates quite a little bit of confusion, but a very, let's say, simple definition is that commutability describes how similar your biological reference material is to an authentic sample. In other words, to what extent does your reference material resemble a real-world sample, right? And in terms of the test, it defines, it's defined by the level of agreement between the test result obtained with your biological reference material and the results that are obtained with an authentic sample, right? So in other words, do the results of your real-world sample match with that of your reference material? To make that a little bit more, let's say, visual, Tanja Dreo made a very nice graph of this, and starting from synthetic nucleic acids test target, so that can either be let's say usually a G block or whatever, going to naturally infested material, it has an increasing level of commutability. The further you go to the right, the more it resembles naturally infected material. So you can have different types and sometimes even some things in between, but it goes from, let's say, clearly synthetic material down to purified material, and that's only a part of your target mechanism. you have spiked material, and that can either be something from a collection, so purified bacteria. You have a healthy plant material. You mix both, and you have spiked material, preferably or sometimes with known concentrations. You can have artificially inoculated material in which you have a healthy plant, you inoculate it with a particular pathogen, and then you use that infected material to produce your reference material, or you can actually just collect material from the wild, and then you get field working material. Again, increasing levels of commutability of reference material. Purity is another, let's say, aspect, right? And basically, it describes the presence or absence of components, including non-target organisms, and when relevant, components of the matrix that could interfere with the results of your test. And basically, they would lead to false positive results. And again, that depends on the test that you are using, the matrix that you are using, and on the pathogen that you are testing for. But it's very important that if you produce reference material and you want to describe the purity, it should be really well documented, right, how that purity was determined. In many cases, the assessment cannot be really absolute. Are you absolutely 100% sure that there are no other target organisms in there, right? To what length do you need to go there? And it can, let's say, be documented or it should be documented in a descriptive manner, right? Like it contains only the target organism, or it also contains material causing possible false positives. People who are going to use that reference material should be informed about, let's say, the purity and about these aspects. Homogeneity is one of the other criteria. And yeah, it sounds really obvious, but if you want to produce and use reference material, one should assume that it is homogeneous, because if not, right, you can have different measurements between or different outcomes in different measurements. So it can introduce measurement uncertainties, and obviously that will affect the interpretation of your test results. That can have an effect on the validation or on your test performance studies or on the proficiency of your lab in certain proficiency tests. That homogeneity can be determined either qualitatively, so just mentioning that the positive controls should be positive, right? Or it can be determined quantitatively, right? And that means that your positive test result should be within a predefined range. And EPO also says very clearly that the homogeneity of your reference material should be determined with the same test as the test that you use, right? Or that the material, that your reference material is intended to be used with. Stability, again, a very obvious characteristic, right? But at least it should be ensured that your reference material will not undergo any significant change throughout its period of its use. And that includes storage and transport. And if there are any restrictions there, they should be well documented and also be provided to the users of the reference material. The only way you can do that is determine that experimentally. And what's often being done is that you use different time points or different conditions, right? And you either use a test that the material is intended to be used with, right? Or a test that will provide general information or information on the general stability. Again, it depends a little bit on the intended use of your material. But as I said before, the stability of your reference material should clearly be documented together with the conditions and the tests that were used to determine that stability. A final or a last, let's say, criterion, also something that is a little bit, let's say, not very clear. It's not a very clear definition. That's something that's called assigned values. more clearly described in EPO, in paragraph 3.6 of the EPO guideline 122, so in the guidelines on interlaboratory comparisons. And there it says that in the plant health field, assigned values correspond to the expected result of the test. And that can be described as a pest is present or absent. It can be described as a certain concentration of the pest. It can be described as a morphological characteristic, right? And it's either a known value, so present, absence, concentration, high, low, right? But it needs to be previously, let's say, assigned. And you also need to have a previously described procedure, right? And the uncertainty around, let's say, the assigned value. So, you know, what's the uncertainty on the expected result of your test? And they also need to be predefined. So we get to the last question of the poll. People work with reference material. Do you actually really produce your own reference material? Yes or no?
So about 80% have answered. I will wait a few more seconds. It's still changing. Yes, and we are at 87% and it's not changing anymore.
Okay.
So I will close the poll and share the results. So 55% of the people answered yes and 45% no.
Okay, so nearly half of the people present actually really produce their own reference material. 45% not. Okay, interesting. Because I would like to discuss, let's say, the process for the production of the reference material in a slightly more detailed form. This is a figure also designed by Tanya, and it's based on the standard operating procedure we developed within Work Package 3. And again, that deliverable is also available at the Work Package 3 page of the VALITEST website. You can download it. And that figure is, let's say, slightly different from this one you see, although it also describes the process of producing reference material with different steps. And the essence of this whole procedure and standard operating procedure is that you have a specific sequence and quality control steps of these steps, and you have to follow them. But the sequence and the steps that are necessary also depend on the biological reference material and its source. In the standard operating procedure in deliverable 3.3, we start with the different sources. So working material, field material, working collection, reference collection, culture collection, and true reference material, going through the steps and so on. And let's say these steps that you need to go through should be determined in the planning phase. And we have this scheme, this process scheme, this flowchart describes these different phases, and I will go through the individual ones now in a little bit more detail. So the first thing to do is actually always identify what your needs for reference material are. What does it need to do? Where are you going to use it for, right? And what are its desired properties? So you have your list of descriptors, which we just discussed, and not all of them may apply to your specific reference material, but you need to, based on these descriptors, you need to clearly describe what the properties of your reference material are. You need to list them, right? So you know in advance what that is required when you really start using your reference material. Once you've done that, you can go to your the planning of your production of your reference material. And that planning includes a risk analysis and a, let's say, definition and defining your, let's say, the points where you need to perform your quality control during your production phase, right? Obviously, identity is probably the first thing to start with, that if you start producing your reference material, you need to make sure that you are actually really working with the correct material, right? But the planning of the production, right, also includes that you need to take into account the source of the material, the processing step you need, then the particular sequence of experiments that you need to perform to determine these properties. You need a plan for that. You need to make an analysis of the risks that are, let's say, associated with all these processing steps, and you need to define in which point during your process you need to check which, let's say, quality points and define your quality assurance. The next phase is that you need to get your components. You haven't actually started producing it yet. You're still in the planning phase. Then you need to get your material. And then again, you end up with, where do you get your material from? Is it from a collection? Is it from other sources? So is it from the field? Is it from a working collection? And so on. And obviously, the first the things that you need to determine are identity and purity. But again, this is just an example. It may depend on your reference material and on the source of your material, whether or not additional criteria should also be determined, right? And if you pass your first quality control steps, right, then you can start, let's say, start your production material and your processing steps. Everything needs to be documented, right? And that should be predefined. Then you start processing your material. You source it, you have identified it, you know that it's fit for purpose at least, and then you start to really go through the processing steps to start producing your reference material. When you've sort of finished your processing steps, or during the processing, you end up in your, let's say, process whereby you actually really determine what your criteria are. And again, this is an example, so the things mentioned in these boxes may not really apply fully to your reference material, but it just shows that obviously there are different criteria that you need to determine. You need to document everything, and you need to cross-check with the things that you have predefined because you have, in your first phase, identified what your properties of your reference material, let's say, should have. When everything's okay, when everything's passed, then basically you end up with reference material, right, with the determined predefined properties, which is fit for the intended use. The final step, because I mean you haven't really finished yet, is that you have to report on its properties. You have to summarize and report all the properties and the processing steps used, and so on and so on. Usually that is done through a technical sheet that is supplied with your reference material to the people who are actually going to use it, and whether we use it in-house or whether you actually provide it to other laboratories, it's not really that important. The information on the processing and on the production and all, let's say, its technical information should be available and accompany the reference material. So, in conclusion, right, we've seen that biological reference material may consist of very different types of material and can come from very different sources. You really clearly need to predefine its intended use, because that will determine what properties are required for that particular reference material. There are different descriptors or criteria that you can use to describe its properties, and these may differ with the type of material. Its production should follow specific steps, preferably in a standard operating procedure, but again, it may depend on the material itself. The standard operating procedure in deliverable 3.3 may serve as a guideline, and hopefully that helps. And very important, documentation of all the material and the process is extremely important, because everything should be traceable, and obviously in terms of your quality control, people should be able to check and get all the information that is needed, that is associated with that material. So that sort of brings me to the end of my lecture, and I think we're very nicely within time. Again, I would like to thank Tanja at NRB in Slovenia, and Anna van Diepingen, who were a really great help and very much involved in work package 3. And you know, I'm happy to take any questions. I think there's a last slide just to let's say show that the next webinar will be on the 22nd of March on Monday. Charlotte?
Yeah, but I guess we can start with the Q&A session first. So yeah, so if you have any questions for René, you can either put your question in the chat, or you can also raise your hand and we can give you the floor. There was one question during the webinar about interoperability. So, but it was a question from Lina, but so I put the question in the chat. What does interoperable means in this context? Maybe Lina, if you want to give more information on your question, I can unmute yourself and you can take the floor if you want. So you will have to unmute yourself if you want to explain your question.
Can you hear me?
Yes, we can hear you.
Yeah.
René, thank you. Great presentation. There was a slide where you used that term for the reference material, and I was wondering if it meant that you could use that reference material under different conditions, labs, methods. Like what did it mean in that slide?
I'm not really sure again what slide that was, but
towards the beginning.
All the way at the beginning.
I should have taken a screenshot.
And other attendees, if you have any questions, so there are no more questions for now. So if you
coming back to your question, Lina, what you said is sometimes reference material can indeed be, let's say, be used in proficiency tests, basically meaning that different labs can use their own method. And then it can be applied in obviously different tests. Interoperable, I'm not really sure in what context we need to see that. I can't find the term anymore.
It's on slide 17, I hear.
17. Which is here, right? Ah, right, yeah. That has to do with the FAIR principles, right? Yeah, that does not really let's say, applied to reference material as such, interoperable is a term that is used in, let's say, data management itself. So it means that data need to be also exchangeable and be as far as I interpret it, be able to be used in different settings, right? So it's not a term that actually applies to the reference material itself. Interoperable in this case means that it applies to, or it applies to the use of the data that, you know, that it applies with the use of the data on reference material.
So I put in the chat the answer from Tanya, and maybe Tanya, if you want to take the floor to explain this a bit more, you can unmute yourself.
Okay, hi, can you hear me?
Yes.
Yeah.
Okay, yeah, this is indeed linked to fair data principles, which can of course be applied to materials as well. So we use them as guides towards what we felt is our goal also with respect to reference material. So indeed, you are correct. We were thinking under this term of having reference materials that can indeed be used in different studies, can feed into different processes, and can be used to assess or to be used as controls for different methods, etc. But it also means that the reference materials are linked with all the necessary metadata that you need later on to compare the results with other studies, that you have enough information to make an evidence-based judgment whether this study is comparable to something that you are doing, etc.
Yeah.
Does that make it a bit clearer, or it just made a bigger mess?
Oh, it makes sense. Thank you.
Thanks.
So there is another question from Maria. So she said the presentation was very good. About the purity, you talk about the false positives and the matrix effect on false negatives. So I think she is asking about can the matrix as also false positive can also be responsible for
false negatives. Yeah,
yeah,
I mean, that is obviously something that can happen, but that is usually more related to, at least in my point of view, related to the type of test that you are using, and not so much to the reference material itself. But I don't know. Tanya, can you also comment on that?
Can you please repeat the question? Sorry.
Well, no, I mean, the question was, is that we, in terms of purity, we talked about the effect that purity can have on false positives, right? So if there's other material there, but Maria also asks about, is there a matrix effect in terms of false negatives? Can the matrix also affect, let's say, the chances on false negatives?
Yeah, that's absolutely true. For example, what we see in case of, if I give an example from Xylella, in case of matrix of olives, of testing olives, we know that there's a lot of inhibitors there, but also there seems to be the effect of polyphenols in the matrix, coming from the matrix, that affect how accessible DNA of Xylella is later on for amplification. So even if we manage to isolate it, it's not accessible for amplification. So that would give rise to false negatives. So yeah, it can affect the result in both ways. It can be false positive and false negatives.
Yeah. But again, that is, in my point of view, that is something that is more related to the actual test that is being used. and not so much, let's say, the purity of your material in terms of presence of your reference material and possible presence, for instance, of other pathogens in there. It's related to other components in the matrix, absolutely true, but not so much to the presence of other pathogens that might be cross-reacting in your test.
So we have no more question. I will wait a few more minutes. So yeah, in case you have a question, do not hesitate to raise your hand or put the question in the chat. So nothing is appearing. So I would like René to thank you for the very nice presentation. And I also would like to thank all the attendees.
Yeah, thank you.
There will be a satisfactory survey at the end of the webinar and in the follow-up emails. So it's really short, so do not hesitate, all of you, to provide some feedback on this webinar. The presentation was recorded and it will be on our website. So do not hesitate to go back to it if you need. The next VALITEST webinar will be on the analysis of TPS results and how to tackle this analysis. It will be on Monday, 22nd of March at 2 p.m. And next week, there will also be a practical training session on how to organize test performance studies. So this is it. I cannot see any other question in the chat, so I will close the webinar. Thank you, René, again, and thank you all for your participation, and see you at the next VALITEST webinar. Bye-bye.
Okay, thanks a lot. Goodbye.